Well, welcome to the second session of the 2021 Brown Bagger webinar series, where our focus is on reducing the environmental impact of beef production. Today, we have two speakers, Dr. Samodha Fernando, who's a professor at the University of Nebraska-Lincoln, who's going to discuss the biology of methane production, and then Dr. Jennifer Bormann, who's a professor at Kansas State University, who's going to give some insights into research they've done relative to the genetic components of environmental impacts. So again, this is the second session of our Brown Bagger webinar series brought to you by the National Beef Cattle Evaluation Consortium. If you've not done so yet, I'd encourage you to visit the National Beef Cattle Evaluation Consortium website. The web address is given here. And on this website, you can find recordings of previous Brown Bagger webinars and also a revised edition, that would be the third edition of the sire selection manual. Today, I'm your host, Matt Spangler. I'm a faculty member at the University of Nebraska in Lincoln, and this webinar series is brought to you not just by myself, but a couple of other colleagues, Dr. Dare Bullock from the University of Kentucky and Dr. Bob Weber from Kansas State University. Our first speaker, as I mentioned, is Dr. Samodha Fernando. Dr. Fernando received a bachelor's degree in Sri Lanka before moving to the US, where he received both a master's and PhD at Oklahoma State. And then he did postdoctoral work at MIT before we were lucky enough here at the University of Nebraska to attract him. He's now a fully promoted professor in our animal science department and works in the area of microbiology. And I'm hesitant to say rumen microbiology because Dr. Fernando works across a multitude of species and is a strong collaborator with a multitude of disciplines, not just within our department, but beyond. And so with that, I'll turn it over to our first speaker, Dr. Fernando.
Thank you very much, Dr. Spangler, and thank you for giving me this opportunity to talk about methane production today. All right, so just to give you an idea of how much methane is being produced and why methane is important. One, methane has the capacity to capture energy 21 times compared to carbon dioxide. And if you think about human-associated factors that contribute towards methane production, actually anthropogenic methane is considered as one of the biggest sources of methane production. So in the US, agriculture contributes about 10% of total greenhouse gas emissions. And if you take feed production removed, livestock contributes about 4% of greenhouse gas emissions. So not a huge amount, but out of that 10%, about 38%, or out of the 4% methane, 38% of the methane is actually produced by livestock species. So oops. All right. So the United Nations Food and Agriculture Organization had this global livestock environmental assessment model. So they went and looked at, okay, what are the sources of emissions and how does livestock contribute towards these methane emissions? So as you can see, North America, the US is not the biggest producer of greenhouse gas emissions. You can see actually South America produces a lot. But what's interesting is in North and South America, the majority is actually of the greenhouse gas emissions are actually coming from cattle production, beef cattle, specifically beef cattle production. And then when you go and look at, okay, per kilogram of protein being produced, how much methane or how much greenhouse gases are being emitted, cattle or beef cattle are only second to actually buffalo meat in South Asia that we see. So what came out of this assessment was that there's lots of opportunity that we can actually reduce greenhouse gas emissions by reducing greenhouse gas emissions from cattle production. So, but even if you think about cattle or any other environment or any other sources of methane production, there's one thing in common. What's common is that this methane is being produced by microbes, or a special group of microbes known as methanogens inside. So there's about 60 different species isolated from different environments, ranging from waterlogged soils to gastrointestinal tracts and landfills and different environments. But when we think about the rumen and the amount of methane being produced, we have a very poor understanding of these microbes, or how these microbes produce so much methane inside the rumen. To date, we have only 5 species that we've actually isolated, and using culture-independent genome binning approaches, there's about 10 different species that we have genomes or information about methane-producing organisms from the rumen. So before I talk about the biology and why methane is being produced in the rumen, just wanted to give you an overview of what is the rumen. Why does a ruminant animal have a rumen? So as you all know, ruminant animals eat this cellulose-rich, highly complex carbohydrates. And if you think about when we feed a ruminant, we are actually feeding the microbes that are inside the rumen. But microbial digestion is slow, and you need more time to digest. And that is one reason that these ruminant animals have evolved this rumen that allows a longer time for the microbes to break down these complex carbohydrates. So the rumen is anaerobic, and there's a huge advantage to the host because of the rumen being anaerobic, and I'll talk about that in a few minutes. But this rumen is so large that it can hold 40 to 60 gallons of rumen fluid and can contribute to about 65% of the total gastrointestinal tract of the animal. So in addition to providing the animal with energy in the form of volatile fatty acids, these rumen microbes actually flow into the small intestines and become a protein source. So we call it microbial cell proteins, become actually a protein source to these animals as well. So what is this rumen microbiome made of? So it actually has all domains of life. It's predominated by bacteria. So if you look at the picture on the top, which I'm showing here, you can see actually a majority of those domains here. So on the back, this is actually a fiber particle. This is the outside of the fiber particle, but you can see this fiber particle is teeming with bacterial cells. And what you see here in large is actually a protozoa. So bacteria inside the rumen accounts for about 100 billion bacterial cells per mil of rumen fluid. And I said there's about 65 gallons of rumen fluid that a mature animal could have. So there's a huge population of bacteria, and majority of these bacteria, as you see in this figure, is actually attached to feed particles, compared to only about 25% of the bacteria is actually free floating. The protozoa is the second largest population, like I said, which is shown here. These protozoa can actually engulf and control bacterial populations. So they can feed on these bacterial populations, so help control certain bacterial populations. But these protozoa on their own can actually depolymerize pectin, engulf starch and fiber particles, and break down fiber particles as well. Fungi population is very small inside the rumen. It accounts for about 5 to 10%. But these fungi are critical because they help break down those or basically solubilize those lignin, so that bacteria can gain access to these mature fibers and break down those cellulose to get energy to the host animal. There are archaea. Archaea are the ones that actually produce methane inside the rumen. And so a group of archaea are called methanogens. Those are the ones that actually produce methane inside the rumen. So they are present around 3 to 5% of the total population. So very small of the bacterial population. So very small in terms of their abundance, but they produce a lot of methane. And there are also viruses that help structure bacterial communities, but we know very little about what viruses do inside the rumen. So if you think about these microbes inside the rumen, you can think of them as a microbial food chain. So all these organisms alone don't have all the enzymes to break any complex carbohydrate. But as a team, they can break these complex carbohydrates down to monomers or substrate that other microbes can use. So similarly, what methanogens actually do is they don't have all the enzymes to break down those complex carbohydrates. What they do is they use byproducts or end products of certain metabolism from other microbes for their metabolism to produce methane. So basically, substrates from certain microbes are a source of input for other microbes to break it down. So later on, I'll talk a little bit about how we can control methane, what are the opportunities or what are the methods available. And one way is sort of controlling the bioavailability of certain substrates for methanogens so that we can reduce methanogenesis or methane production inside the rumen. So a few minutes ago, I mentioned that the rumen is anaerobic and it is advantageous to the host because the rumen is anaerobic. So in our cells, if we get a glucose molecule, glycolysis will break down that glucose molecule to pyruvate, and then using the TCA cycle or the Krebs cycle, we'll break it down all the way to carbon dioxide and water. So we can get the total energy that's in that glucose molecule. But because the rumen is anaerobic, the microbes can't get all the energy from that glucose molecule. So what happens is the microbes, they use glycolysis to make pyruvic acid or pyruvate, and then they produce these VFAs that are energy-rich compounds that the host can absorb. So if the rumen was aerobic, the microbes would break it down all the way to CO2 and water and get all the energy, so the host won't get any of the energy. So the rumen being anaerobic is critical for the host so that it can actually get that energy from the host. So inside the rumen, there are multiple reactions happening where VFAs are being produced. So like glycolysis, which I just mentioned a few minutes ago, takes glucose and produces pyruvate. So what I want you to bring your attention is to this hydrogen. See, there are certain reactions like acetate and butyrate. When it's produced, you can see these hydrogen molecules that's been released. These hydrogens are substrates for methanogens that they can combine this hydrogen with CO2 to produce methane. There are other reactions like this propionate, C3. So propionate, as you can see, it actually uses hydrogen as it produces hydrogen. So it's a hydrogen sink. We call it a hydrogen sink. So it actually acquires hydrogen when it produces propionate. So what that means is it's taking substrates away from methanogens so to produce propionate. Therefore, one strategy people are using to reduce methane is, can we increase propionate production inside the rumen? And I'll talk a little bit more further down as well. So if you think about this total hydrogen being produced inside the rumen, about 50% of that hydrogen is actually going into methane production. And part of it, like for propionate or succinate production, they utilize hydrogen. So part of it goes to VFA production. The rest is actually going to, what I mean by others here is actually for biohydrogenation. So when you have an unsaturated fatty acid that enters the rumen, because the rumen is anaerobic, there's no beta oxidation happening. So what happens is rumen microbes can actually unsaturate that hydrogen, saturate the unsaturated hydrogen. So the double bond can be converted into single bonds by microbes and utilize hydrogen to do that. So what we've seen, and here's data showing that as you increase acetate and butyrate production, or as you increase VFAs that produce hydrogen or release hydrogen, you are actually increasing methane production. So in this graph, is C2 and C4 is when you have acetate and butyrate. But as you increase your propionate production, what you're seeing is this ratio goes down and you have less and less methane being produced. So propionate production is actually competing with methanogenesis for hydrogen, and therefore you can actually reduce methane production by increasing propionate production inside the rumen. So the question is, why do the rumen have methanogens? Or what is the role of the methanogens inside the rumen? So I mentioned earlier that inside the rumen, glycolysis happens, and this is on your right-hand side, what you're seeing is glycolysis. So your input is your glucose molecule. You end up with a net production of 2 ATPs, but you also produce these molecules of NADH. So NADH, so for glycolysis, you need NAD. So if this NADH has to be recycled back to NAD so that glycolysis can go forward. Because this pyruvate is the substrate for VFA production. So if you don't have glycolysis going on in the rumen, then the rumen function will stop because you need that NAD equivalence to keep glycolysis going forward. So what methanogens do is actually take this NADH that's been produced in either glycolysis or the certain VFAs like acetate or butyrate production. They have these enzymes called hydrogenases, and they convert that NADH back to NAD so that this NAD is available for glycolysis. So what happens if methanogens are not there in the rumen? There's two ways you can, there's multiple ways you can recycle this NADH. One is you can use some of the pyruvate that's been produced by converting it to lactate or ethanol. When you convert pyruvate to lactate and ethanol, you're not getting any ATP, whereas when you convert pyruvate to acetate, the microbe is getting ATP and you're getting VFAs that goes into the host. So if you don't have the methanogens, you're actually wasting energy because of pyruvate just to recycle NADH, making the animal or the rumen ecosystem less efficient, because now it's wasting energy that could be used for the host for either milk production or meat production, just to recycle NADH, making the rumen less efficient. But another way you can recycle is to succinate or propionate. So if we can recycle NADH through succinate or propionate production, you can actually increase the energy going into the host and also increase rumen function. So the methanogens actually play an important role inside the rumen by recycling this NADH back to NAD so that glycolysis can move forward. The other thing that they do is they actually decrease the partial pressure of hydrogen inside the rumen, because these hydrogenases that convert NADH back to NAD, even in reactions such as succinate and propionate production, want low partial pressure of hydrogen. So by these methanogens help recycle NADH back to NAD for glycolysis, but also help maintain a low partial pressure so that these hydrogenases, the enzymes that actually recycle these molecules, can be active inside the rumen as well. So there is this tight relationship between these hydrogen producing organisms or reactions that produce hydrogen and hydrogen utilizers, such as, so when you co-culture these bacteria and methanogens together, you actually see them both grow well in co-culture or in environments. So inside the rumen also, these relationships actually help. So certain microbes actually enhance the growth of methanogens inside the rumen. Another thing is these protozoa and fungi have these unique structures called hydrogenosomes that produce large amounts of hydrogen. So actually, these increased amount of hydrogen in production from fungi and protozoa is associated with methane production, and you can actually see methanogens attached to protozoa cells just because of the hydrogen being produced. So one strategy scientists have tried in the past is, can we actually defaunate or kill all the protozoa inside the rumen so that you have less hydrogen being produced from protozoa so that we can control methanogenesis or reduce methane production inside the rumen. So it's not only carbon dioxide, hydrogen that goes into methane production. There's other substrates such as methanol, acetate, methylamines. There are multiple substrates that can be sources for methanogenesis, but inside the rumen, it is considered that 80 or 90% of all methane being produced is coming from either formate or CO2 and hydrogen inside the rumen. So just to give you an overview, I'm not going to ask any biochemistry questions at the end, but just to give you an idea of what methanogenesis happens and where each of these substrates come in. What is the critical step of methanogenesis is this last step of coenzyme M or coenzyme M reductase reaction. That is where, because it is common to whatever substrates comes into methanogenesis, whether it's formic acid, CO2 or hydrogen, methanol or acetate. So some of the strategies have been to reduce methanogenesis by attacking this last step of methanogenesis. Sorry. So there's some of you might have heard about 3-NOP, which is actually a substrate that actually attacks this reaction and binds to this coenzyme M so that methane production can be inhibited. So how can we control methanogenesis or reduce methanogenesis? So there are some studies or several studies looking at what's the effect of genetics and the microbiome on methanogenesis. So this study by DeFord and others actually looked at host genetics and the microbiome, and they showed that there's about 21% heritability of host genetics affecting methane production and another 13% from the microbiome. But the interesting thing they found is that the heritability or the genetic component that affects methane and the microbial component that affects methane is different, and therefore they can independently be targeted to control methanogenesis inside the rumen. Then later on, there was a few studies that actually showed that the host genetics actually select for certain microbes that in turn influences methane production. In this case, these two studies showed that actually host genetics is not directly affecting methanogenesis, but it's affecting methanogenesis by selecting for certain microbes inside the rumen ecosystem. So if we are to think about how we can actually control methanogenesis or decrease methane production inside the rumen, sorry, my slides are inside the rumen. So either we can use inhibitors that decrease the production of hydrogen or CO2 or formic acid, or we can find alternative hydrogen sinks. I'll talk about nitrates and sulfate in my next slide. Or like 3-NOP, we can target methanogenesis pathways to reduce methane emissions. So here, to give you a summary of the strategies that's available and what's been used today. So there's feeds, like I said, there's a microbial food chain that would break it down and produce VFAs. In the process, you produce as byproducts CO2 and hydrogen. So archaea or methanogens can use that CO2 and hydrogen to produce methane. But there's another group of microbes inside the rumen called acetogens. These acetogens actually can use hydrogen and CO2 and produce acetate, which would be beneficial. But the problem is that this these acetogens need very high partial pressure of hydrogen, and we don't reach those high partial pressures of hydrogen inside the rumen because of methanogens. So although these acetogens are there, they don't play a huge role in producing acetate. So dietary intervention using sulfates and nitrates have been tested, and because you could use sulfate or nitrate as alternate electron acceptors, terminal electron acceptors to compete with methanogenesis. But the problem is it produces hydrogen sulfide, which is toxic to the animal. And in the case of nitrate, you can have nitrite toxicity because nitrite could get absorbed, bind to your hemoglobin, and the hemoglobin can no longer bind to oxygen. So therefore, you could actually the animal could die of nitrite toxicity, but you can adapt the animal by slowly increasing levels of nitrate to sort of have an alternative to methane production or an alternative electron acceptor. Right now, the most viable and strategy that people are going after or scientists are going after is this propionate or succinate production, because it also gives energy to the host as well. So, so a few years ago, we had a study where we used a Kale and Gale system, but when the animals come in to actually feed, we actually collected methane samples and predicted how much methane these animals are actually producing. We could see that low and high quality forage, there was a difference in methane being produced. We wanted to go and look at, okay, how does the microbiome affect this methane production? Is this high and low quality for animals, forage fed animals, which show different methane? As you can see, what's interesting is we didn't see a difference in the methane producing microbes. But what we saw is these high and low methane producing animals had differences in bacterial diversity, the microbes that actually produce some of those substrates that feed into methane production. We also went and looked at their function, and you can see functionally their pathways and genes and pathways were also different in this high methane producing and low methane producing animals, showing that these pathways of these bacteria that produce substrate for others, for archaea, affects methanogenesis. So just a few key points to end up. Methane production plays an important role in making sure that the animal is efficient in energy production, or the microbiome is efficient in energy production for the host animal. But so any strategy that we use should think about how can we recycle NADH, because if we just kill the methanogens, because we are going to use some of that pyruvate that could go to VFA production just to recycle NADH, we are making the rumen inefficient. So we need to find ways that we can maintain that recycling of NADH while reducing methane production. So dietary intervention, microbiome manipulation, and genetic selection are viable strategies to reduce methane production in the rumen ecosystem. With that, I'll take any questions. Thanks.
Thank you, Samodha. If there are any questions for Samodha, feel free to type those in the chat box now, and we can maybe handle a couple of quick ones. Otherwise, Samodha, if your schedule allows, if you stick around to the end, we could handle some questions then.
Yep. Matt, I'm planning to stay till the end, so we can actually visit about them together at the end too.
Great, thank you. So our second speaker then is Dr. Jennifer Bormann, who I mentioned is a professor at Kansas State University, where she has both a teaching and research appointment. Dr. Bormann, a native of Iowa, actually completed undergraduate work at Iowa State before going to Oklahoma State to work on a master's degree. And then she came back to Iowa State to complete a PhD before moving to Kansas State as a faculty member. And Dr. Bormann's going to give some insights into some research relative to genetic components of environmental impacts. And so with that, Jenny, I'll turn it over to you.
Okay, I think I have control now. Thanks, Matt. And thanks to the group for the invitation today. I was asked to talk a little bit about looking at different genetic components to some of the sustainability traits and what the environmental impacts are genetically, perhaps, that we can control in beef production. Just want to quickly acknowledge the team here at K-State, Dr. Weber and Dr. Rolfe, as well as our graduate students, because as you guys know, if you have good graduate students, you don't have as much work to do because you can use a lot of their work. So I definitely borrowed very liberally from some of their stuff for some of today's content. So I kind of decided to look at 3 different areas of environmental impacts and look at what we know about the genetic components of those. So I'm going to talk first about methane production. So thanks, Dr. Fernando, for the lead into that. And then a little bit about water use and then this idea of adaptability. And then Dr. Place's talk last week, which I thought was excellent, toward the end of the Q&A, we were talking about maybe some things that we already have that we could use to do a better job of improving our sustainability and our environmental impact. So I'll finish up with a little bit about some existing tools that we might do a better job of using to help with the environmental impact of beef production. Okay, so methane production. Now, I'm not going to spend any time talking about why it's important because I think we've heard that already today and of course last week as well. The problem with methane, obviously, is that it's really, really hard to measure on an individual animal. We talk about things like feed intake being hard to measure, but I think methane really kicks it up a notch. It's just really hard to get good measurements. Now, there's been some work on using predicted methane, so using dry matter intake and diet and things like that to predict an animal's methane. And those prediction models do a pretty good job on average. So they can do a good job of predicting what a group of animals might produce or what the average in a group of animals might produce. And that's fine if we're just looking sort of at gross measures of methane production. But for genetic selection, we don't want the average animal, right? We want to find those animals that are outliers. So we need to find animals that produce less methane independent of the predictor variables that would go into a prediction equation. And so another way of saying that is within a contemporary group, basically the animals will rank the same for methane production as they will for dry matter intake based on prediction models. So that doesn't work. Now, maybe a side note is that if we do decrease dry matter intake as a component of a selection program to increase feed efficiency, that will decrease methane as well because the dry matter intake and methane production are highly correlated. So measuring methane really for genetic selection is critical. We have to have methane measurements on individual animals to find those outliers. But it's hard. So there's a couple of different ways. The gold standard would be the respiration chambers, where basically an animal stands in a box and they measure the air in and the air out and figure out how much methane is produced. That would be this top picture of several chambers in a row. Obviously, that has huge impacts on animal behavior, animal performance, feed intake. It's obviously a very unnatural situation for that animal, and so that's going to affect their methane production as well. And it's really pretty much impossible to measure enough animals for a genetic evaluation, get enough records on enough animals. There's another technique called sulfur hexafluoride tracer, where there's a bolus of sulfur hexafluoride placed in the rumen, and it releases gas at a known rate, I think. And it's hard to see on the picture, but there's a little tube on this halter on this animal's nose, and so that tube is capturing methane as well as other gases as it comes out of that animal's respiratory tract. Again, very labor intensive. It can only be used really on a small number of animals for a short amount of time. Difficult to get enough measurements to do a good job of a genetic evaluation. Some newer technology are the open circuit gas quantification systems. And these are systems that can they bait the cattle to stick their heads in what's really kind of a fume hood. And then as they're sitting there eating, it measures the gases that those animals are respirating. And so the big advantage with this kind of system is, as you can see, it can be set out in a production type situation. These animals, you can be grazing native range or you can put it in a feedlot, but it is much more of a natural production system. So you're not affecting the behavior and the intake as much. And there's quite a few of these out there now. Several universities have them, research centers. And so I think there's some promise that over the next few years, we can start to get methane measurements on large enough numbers of animals to maybe make a difference. But if we look back at what's been done in beef cattle, there's not been much, but there's been a little work on genetics of methane production in beef cattle. Early on, the Australians were really kind of the leaders on this. And so what they sort of looked at first was in their selection lines for RFI. So they've been selecting for RFI for quite a while in Australia. And they were looking at methane production of cattle with different levels of RFI. And so one of the first studies was basically just comparing high and low steers for RFI and their methane production. And they did find that by reducing RFI, 1 kilogram per day reduction in the breeding value for RFI actually was associated with over 13 grams per day reduction in methane production. And if they divided those cattle into high and low groups, the low RFI steers did produce quite a bit less methane than the high RFI steers. And they went ahead and simulated this and showed that if Australia continued to select for RFI, there would be an associated improvement in methane production of about 3% over 20 years. So small improvements, but certainly makes a difference. The Canadians did a similar study, and I think what's interesting about this one is that the numbers come out pretty similar. The low RFI steers were about 28% less methane producers than the high RFI steers. So very similar results to what we saw in the Australians. And they showed that resulted in about 16,000 less liters of methane per animal for those low steers. What was also interesting about this study, and they only looked at phenotypic correlations, but really not much relationship between methane production and growth rate, which is good news because maybe we can find cattle that can grow fast with less methane. So kind of the biggest study, most comprehensive study in this area was done in Australia over the last few years. And they were able to measure over 1,000 Angus cattle in the respiration chambers. That picture I showed a minute ago with the rows of respiration chambers was from Australia. And so they measured 10 animals at a time over many, many years, replicate after replicate, to get to 1,000 animals with actual methane production data. And so from that analysis, they showed that the heritability was about 27% of methane production. And so that, a low to moderate heritability, we certainly can make change if we've got the data with a heritability of that level. They also looked at some other traits. And that's, I think, a really good question to think about is if we are going to select on methane, what's the appropriate trait for selection? We conceived by the genetic correlations that methane production, again, highly, highly correlated to dry matter intake. And so if we select for decreased methane, we're going to decrease dry matter intake. They looked at methane yield, which is a ratio trait. That's methane production divided by dry matter intake. And so obviously that would be much more independent of dry matter intake. We know from lots of other work that selection on ratio traits can be problematic for a couple of reasons. And then obviously they do a lot of residual feed intake work in Australia, so they also looked at residual methane production by several different equations, actually 4 different prediction equations to predict the expected and then calculated the residual. And so all of them ended up with a heritability of about 10% and more uncorrelated to dry matter intake as you would expect. What's also interesting is the genetic correlations of methane to our other traits. Again, if we think about making selection on methane, what does that do to our production traits? And obviously just the raw methane production, amount of methane produced per animal, highly, highly correlated to intake and size. Bigger animals produce more methane. I don't think that's, groundbreaking to anybody. Some of their other traits by design are less correlated. So methane yield, the ratio trait, within the range of standard errors, pretty much uncorrelated to the growth traits. And the same with the residual methane production equations. And obviously that varies depending on which of those equations you're looking at, but much more uncorrelated. When we look at carcass traits, we see similar results where methane production is maybe a little bit genetically correlated, especially to ribeye area and IMF. We think about those maybe as related to size of the animal, perhaps. That makes some sense. But then residual methane and methane yield, again, would be less correlated. So the conclusions kind of from this first part of that study was that we had low to moderate heritability on methane production and methane yield, so we could make progress with selection. And they concluded that we shouldn't select on raw methane production because of those unfavorable genetic correlations with other traits. What they didn't mention, and the question I would have is, what about a selection index approach where we put methane into a properly weighted selection index? because we know that there's problems with ratio traits and residual traits. They did some continuations of this study where they did the genomic work. So all these cattle, these 1,000 head of cattle with methane productions were also genotyped on the 50 or 700K, and did some genomic analyses, came up with about the same heritability, which is good. Accuracies of the breeding values were somewhere in the neighborhood of 0.3. A little bit depends on the methodology used. But I thought what was really interesting was the amount of the genetic variance in those methane traits that was explained by the SNPs. And so, again, relatively low to moderate heritability, but of that genetic variance, nearly 70% was explained by the SNPs. Methane yield about 60%. So what that says is that there is some potential here that genomics could really help us in doing selection for methane, which is great because it's a hard trait to measure. So really some potential there. Another continuation of this study looked at a different trait. So a different ratio trait called methane intensity, where we scale methane by the body weight, not the dry matter intake. That comes out, about the same in heritability. Very highly correlated to the other ratio trait, methane yield. So I'm not sure exactly, how much that helps us. They did publish 165 SNPs that were significant for methane production. But what's interesting about this one is they also measured methane on a Holstein population and then tried to compare the two and found that not surprisingly, that there were different SNPs that controlled methane or predicted methane production in those two different breeds. And I think that's probably not surprising because we've seen that in genomic selection for other traits as well. And so we'll have to be careful as if we develop genomic tools for methane production to really think about the populations where we develop those tools. So kind of some general conclusions from the methane work is we know that methane is heritable. We know that it's highly correlated genetically to dry matter intake. There are some other traits, methane yield, methane intensity, residual methane, that might be candidates for selection because they're less correlated to those production traits. And genomics appears to be very promising as a way to help us make selection for methane production. I think some of the questions we're left with is, first of all, what measurement is the most useful? Is it production? Is it yield? Is it intensity? How do we quantify methane production for selection? And if we think maybe selection index might be a good way to go, that means we need an economic value for methane production. So I think that's going to be a challenge. There's certainly a direct economic value because, as Dr. Fernando showed, it takes energy to make methane. And so part of the feed energy that you're providing to cattle is wasted in methane production. So there is a direct benefit to decreasing methane. But certainly there's maybe an indirect benefit to greenhouse gas emissions. And so, you hear about things like carbon credits and methane tax. And so it's definitely not inconceivable that there may someday be a very direct value for cattle that produce less methane. But I think that's certainly a question that's yet to be answered. Okay, so let's talk real briefly about water use. And it's brief because there's been very, very, very little work on water intake in beef cattle. There's been more work in mice. We know it's heritable in mice. We know that there's breed differences. Different breeds and breed crosses will drink different amounts of water. But there's really been not much work done on the genetics of water intake in beef cattle. So there was a study done at Oklahoma State over the last few years that was measuring water intake on about 580 steers. And based on this data, Alberg and others calculated that the heritability of water intake was about 40. So pretty highly heritable trait. And obviously standard errors are larger here because of the smaller study, but appears that there's a decent amount of heritability for water intake. I think what's really interesting about this study, again, is that correlation with gain. Basically no relationship between water intake and growth rate, and even dry matter intake. Pretty low correlation genetically of dry matter intake and water intake. So really what I'm taking away from that is that there are cattle that can eat and gain on less water. Those don't seem to be tied together too much. We also could calculate residual water intake to compare to residual feed intake. Again, highly correlated to water intake, but more uncorrelated as well to gain and dry matter intake. Also looked at the relationship of water intake to carcass quality, carcass characteristics, and again, high standard errors, but really not much there for genetic relationships between carcass traits and water intake. So it does seem, again, that there are animals that can produce good carcasses on less water. So really, again, not much done in this area, but some general conclusions is that yes, water intake is heritable. We could change that through selection if we wanted. That low genetic correlation tells us that there are animals that can eat and gain and grow and do it on less water. And the same with carcass traits. So kind of the questions that we're left with here is, yeah, it seems like there is potential. Can we make genetic change in water consumption without affecting other traits? And this preliminary data would say, yes, we probably can. But is there a value? Similar to methane production, what is the value of selecting and producing animals that use less water? Now, certainly that's going to differ, I would expect, in different parts of the country. And we know that the animal's individual water intake isn't the largest use of water in the beef production system when you think about things like irrigation for feed and things like that. However, maybe there is value in some areas of having animals that drink less water and still are able to perform. And so that might be something we need to think about. I want to move into talking a little bit about adaptability and this idea of adaptability. And some people call it phenotypic plasticity. And really that's just a way of saying that an animal is able to maintain their performance when the environment gets tough. Okay, so can an animal maintain good performance across a wide range of environments? Those animals that can would be classified as adaptable or phenotypically plastic. And so kind of the one way that we looked at this over the past few years is you look at the genetic correlation between the trait measured in different environments. And so this would be sort of our classic G by E, genotype by environment interaction that we learned in Animal Breeding 101, is that you ask yourself, do these animals or these genotypes rank the same in different environments? And so this figure would just be an example where we have pretty significant G by E. Those animals perform differently in different environments. And so there's been some work on this over the years. Back in the 80s, the Georgia group used limousine data and looked at the progeny of sires raised in different regions of the U.S. and looked at the genetic correlation between birth weights in different regions and weaning weights in different regions. And for birth weight, they found the average genetic correlation in different regions was about 0.8, so decently high, but the range was anywhere from about 0.5 to 1, meaning that there were different regions where there was significant sire re-ranking in birth weight. Weaning weight was similar. The average genetic correlation was about 0.7, so some re-ranking with a large range. And so basically, again, that means we've got some G by E. We've got some sires that do better in some regions and some sires that do better in others. Similar analysis in Red Angus with stayability found similar results, but maybe even a little bit lower, that depending on which two regions you were comparing, the genetic correlation was as low as 30%. So significant re-ranking of sires with their progeny in different regions. More recent study looking at weaning weight in Angus cattle, a little bit higher, around 85% genetic correlation. So we definitely have differences in animals in different regions. Another way that we can look at adaptability is to model performance over different environments with random regression. And the Georgia group worked on this a few years ago, looking at weaning weights and yearling weights in Angus field data over different levels of heat stress. And so some of the conclusions they found were that actually heritability decreases when traits are measured under heat stress. Okay, so heritability of weaning weight under heat stress is lower than heritability of weaning weight when measured under a more neutral environment. But they also found positive correlations between the slope and intercept of the random regression. And basically what that means is that animals that were higher performing at a neutral environment or a stress-free environment, if you will, they also tended to be higher performing under stress. And so there was not as much re-ranking as maybe we might think from the previous studies. However, they did note that at the very extreme levels of heat stress, you did start to see some more re-ranking of animals. Will Schaefer in our group continued the work from Alberg and those feedlot steers with water intake. And so after the cattle were kept for 70 days, given ad libitum water, they were gradually stepped down and restricted in their water intake. So they were only allowed to drink 90% of what they would have drank at ad libitum, then 80%, then 70%. Gradually stepped down until they were only being provided with half the water that they drank in the previous feeding period, so half of ad libitum intake. And so this is a graph that shows at 0% water restriction, which would be ad libitum, up to 50% water in restriction, what happened to dry matter intake. And not unexpectedly, we see that it definitely trends downward. On average, animals ate less when you restricted their water. When you look at the genetic correlation between dry matter intake at ad lib, which is 0 restriction, and then 25% and 50% restriction, you can see that the correlation between 0 and 25 is pretty high, seems to be the same trait. But when those animals are subjected to 50% water restriction, the genetic correlation is only about 0.78. So we definitely had some re-ranking of animals as far as their dry matter intake goes when their water was restricted. And just like the Georgia group, we found that heritability did go down under that environmental stressor. And what's interesting is that it's due almost entirely to a decrease in the genetic variance. Okay, so the genetic variance decreased by almost 50%, which then, of course, means the heritability is going to go down. Using that same data before the animals were water restricted, so in sort of that baseline period when they had ad libitum water, we also looked at temperature humidity index. So it's obviously a combination of both the temperature and humidity that indicates heat stress. And looking at the range of THI values and looking at dry matter intake, that didn't seem to change as much. There's maybe a little negative slope, but for the most part, on average, the animals ate about the same under high THI as low. However, there was significant re-ranking. Okay, so again, this is dry matter intake measured at no heat stress and higher levels of heat stress. And the genetic correlation was only about 40%. So lots of re-ranking of animals for dry matter intake as heat went up. Very similar story to what we saw before is that as THI went up, heritability of the trait decreased. Okay, again, genetic variance is driving that. We see decreased genetic variance of dry matter intake at high THIs compared to low. And these are the random regression parameters. And so basically, this is the random regression of dry matter intake on either water restriction or THI. And as we saw in the graph, there's a very negative slope. As restriction increases, dry matter intake goes down. And a much less dramatic slope for THI. We saw that in the graphs. But this correlation is interesting. And so that negative correlation, really what that's saying is that animals that perform really well under no stress take a bigger dive under heat stress. Okay, so that there's an inverse relationship between performance and how they're able to maintain that performance under heat or water stress in this case. So from these random regression studies, we see that in general, it seems that heritability goes down as stress goes up. And at least in the more recent study, it does seem that the higher performing animals are more sensitive to environmental changes. They lose performance more quickly as the environment gets more stressful. And so really that leads to a couple of questions, that if heritability truly is lower under high stress conditions, does that limit our ability to make selection under those conditions, right? Lower genetic variance means lower response to selection. Again, that's animal breeding 101. So is that a problem? And then again, are these higher performing animals selected for high performance under good environmental conditions, are they truly less adaptable and are they not going to be able to perform at higher levels of environmental stress? Okay, so I just want to finish up today and talk a little bit about some of the tools that we have and how we might make better use of them to actually decrease the overall environmental footprint of beef cattle production. And one thing that this group has talked about for years has been matching cows to the environment, finding the right cow to match the feed and other resources that exist in those environments. And there's been some recent simulation work by a student, Drew LeCamp, who's now at University of Nebraska, using a simulation model, inputting things like forage type and rainfall and irrigation use and supplemental feed and all kinds of inputs to figure out what's the most efficient kind of a cow as far as environmental footprint goes. And specifically looking at cow size and lactation potential. And not surprisingly, large cows required more land, right, because they eat more. High lactation animals required supplementation to maintain reproduction, so they were going to require more land and irrigation use for supplemental feed sources. And that really drives the total water usage by these herds, that cattle with high lactation potential need more supplementation, and so we're going to require more water use to irrigate to produce that supplemental feed. And not unsurprisingly, tying back to what we've already heard today, is that those larger cows eat more forage, lowly digestible forage, and so they do produce higher amounts of methane. What's also interesting is then to scale that by weaning weight, because we certainly anticipate a larger framed, heavier milking cow will produce more pounds of weaned calf, more saleable product, and so we want to take that into account. And even when doing that, we see that, well, in this case, the smaller and high milk cows are actually more efficient as far as just grazing land and drinking water, and because they're small, they produce less methane. If those cows are lower milk, then we don't have the supplementation issue, and they do use less irrigation and less cropland. But in general, the large cows did have the largest environmental impact per pound of calf wean. So even though those big cows are weaning more calves, they're using more resources and producing more methane to do it. And so is this a tool that we potentially can use to model what is the best cow, the optimal cow, for different scenarios? Another thing that it's simple but not easy, I guess, would be how I describe it, is doing a good job with our crossbreeding programs. And this is something that our friends in the swine and poultry industry have done a great job of, and maybe we haven't done as much as we could have. But when we think about matching our cows to our environments, maybe the ideal cow in a lot of environments is really not the same as the ideal market progeny. We think about things like growth and carcass traits and efficiency. And so can we do a better job of separating out the sire selection for maternal sires to produce our heifers and then terminal sires to produce market progeny? And we know that the best way to do this is using that static terminal crossbreeding system that our swine friends use so successfully, where you outsource those F1 females that are highly selected for maternal characteristics, only have terminal sires on the place, and sell everything to the feedlot. And that works great if you have a source of those F1 females. And that's a challenge, right, in the cattle industry. It's not as easy as just picking up the phone and ordering 100 F1 heifers to be delivered next week. We just don't have that structure. So another system that could work is sort of a hybrid system between rotational and terminal called the Roto-Terminal System. But even then, we can separate out those mating decisions. If we breed roughly half our cows to that maternal sire that's highly selected for fertility and stability and the traits that are making a difference on the cow side, and then only keep heifers out of that sire, we can breed the other half of our herd to our terminal sire to produce those very high-value market progeny and overall make the system more efficient. Now, obviously, something like AI and sex semen makes this work even better. If we can breed fewer of our cows to produce heifers, that means more of our cows are actually producing a profitable offspring that year. So some technology can make this work even better. But again, that allows us maybe to be a little more efficient in producing our females versus our market progeny. And then I think we mentioned this last week at the end of Dr. Place's talk, is that, those open cows are a killer. And, if you go online and search things like the SPA data and the CHAPS data, obviously there's a wide range year to year, but depending on kind of the region, the year, weaning rate is somewhere in the neighborhood of 80 to 90%. And so basically that means how many calves did you wean for every cow that you exposed the year before. And if it's somewhere in the neighborhood of 80 to 90%, that means it's somewhere in the neighborhood of 10 to 20% of the cows don't produce any beef. And so think about the amount of resources going into those cows, the amount of water, the amount of land, and the amount of methane those cows produce for no human edible product. Okay, so that seems like a place that we can really make a difference, improving that reproductive rate. And then also stayability. Okay, again, if we think about replacement rate or culling rate in the cow herd, it's going to vary, but somewhere in the neighborhood of 15 to 20% is probably close to a national average. Well, if you're culling 15 to 20% of your cows every year, you need to keep that many heifers back. Well, that bred heifer takes a whole lot of resources before she produces any product, right? 2 years before she calves, and actually 3 years before that first calf is actually saleable, human consumable. So that's a whole lot of resources going towards not much beef. And so if we could decrease that, keep those cows in the herd longer, have a smaller proportion of our animals in that developing heifer pool, that's another way to more efficiently use our resources. So again, we've got good tools. Maybe they could be better. I'm not going to argue they're perfect, but we certainly have tools that we can make genetic improvement and we can match our cows to our environment in a better way that's going to decrease the environmental impact. So cow size, lactation potential, doing a better job of utilizing crossbreeding systems, use the EPDs and the indexes that we have for reproductive rates. Certainly there may be better ways to do those, but we do have them. And then also let's think about what are some other technologies that we have? What are things that exist that maybe with a few tweaks could be deployed to make real progress in this idea of an environmental impact of beef production? And because there may be things out there that are relatively low-hanging fruit. So with that, I would be happy to answer any questions, or Dr. Fernando can jump back in here as well, and we're right about on time.
Great, thanks, Jenny. If there are any questions from the audience, please again use the chat box. Type those in. Listening to both your talks, Samodha, a key take home for me is one, reducing methane isn't a zero-sum game, and methane production really is a confluence of not just inherent differences, but also nutritional regime, management, et cetera. And Jenny, really appreciated your comments on indexes. And as you said, the crux is what is the economic signal for reducing methane or any of the trait complexes that you mentioned. And of course, your points about the low-hanging fruit, the tools you already have available is very well taken. So just a reminder to the group that our third session is a week from today, October 20th. And your host will be Dr. Dare Bullock from the University of Kentucky. Two speakers, Kelly Ritalik, who's the president of Angus Genetics, Inc. We'll talk about implications and genetic evaluation of hair shedding. And then Dr. Tad Sonstegard, who's the president and CEO of Excelogen. We'll talk about breeding tools for breed outcrossing and impact on sustainable production for tropical climates. And so if anyone's familiar with Dr. Sonstegard's current role at Excelogen, you'll know that there'll be certainly some mention of gene editing in that talk. So encourage you to tune in next week. And then again, a reminder that recorded sessions from not only this year, but previous years will be available at nbcec.org. So with that, given we're a few minutes over for the day, I'd just like to thank again both of our speakers. I think really outstanding job and covering a lot of ground relative to the basics of methane production, but then also a glimpse into some research efforts for a wide array of trait complexes that might impact sustainability. So appreciate that. And thank you for all of those that attended today. So with that, I'll sign off. Thanks again.